we didn't blast our file because it would take too long. So if you go to the eClass page under DNA Master Resources, you'll see a file that says Octavius Rex blasted file. And you're going to go ahead and you're going to download that file. You can see this is a DNA Master file. And just like before, I'm going to put this on my desktop so that I know where it is. So you can copy it and paste it on desktop. You can move it, just, and you can leave it wherever you want. Just have it somewhere where you know where it is. I'm going to go to DNA Master, and I'm going to open here an archived DNA Master file. So from this point forward, anytime you open a DNA Master file, you're going to pick this choice here, Archive DNA Master File. And I'm going to go to my desktop, and I'm going to choose the Octavius Rex blasted file. I'm going to open it. And it should look, let's make this one smaller, just like you left it before. So I'm going to go ahead and I'm going to save this. And you can save this on your jump drive. I'm going to save this, just on my desktop or wherever. But you should save this on your jump drive. I'm going to save this as Octavius Rex annotated 2.8. So this is the file that we're going to use to start our annotation. Now, we left off in our annotation guide at page 39, where we blasted our file. And they, on page 39, they talk about the blast results. We will get to that when we start using that data. We're going to kind of skip ahead in the annotation guide at this point. So chapter 5 starts on page 41. You really don't need to know much about chapter 5 because we don't use some of these tools we don't use. The tools that we do use, we will come back to in chapter 5. Chapter 6 talks about a couple other tools that we will use. One of them is called Famerator. It also talks about a program called Starterator. So if I pull up the annotation guide, so we did chapter 3, that was importing our FASTA sequence, and we did chapter 4, auto annotate. And again, this is all things that we've done so far. This is the documentation. Looking at the sequence, we went over the frames. We talked about BLAST. This is the end of chapter 4 here. Again, in chapter 5, so of the annotation guide here, we're not going to use most of this, so you don't have to worry about this. Chapter 6. So chapter 6, the first part of this chapter talks about this program called Famerator. We will use this, but we're going to talk about it once we start annotating our genes. So I'm going to skip over that. You can see there's a good amount of information for that. This other program is called Starterator, which we will also use. Again, but this we're going to skip over until we get to the point of using this particular tool. This brings us to chapter 7. So I do want to look at chapter 7 real briefly. So chapter 7 is kind of a guide. These are the guiding principles of genome annotation. And it'll give you an overview of this, and I would like you to read this. It talks about how we use some of these tools and how DNA Master predicts a good number of the genes correctly, but it's not always right. And then there's these guiding principles on page 64 and 65. So page 64 are principles that are generally more important than those on 65. So I would like you to look these over. We will come back to these as we're annotating and apply them to our annotation, but these are kind of rules that we go by when it comes to our annotation. So when we're predicting genes, these are things that we want to take into account. There are always exceptions to rules in biology, but these are a good guideline when you're trying to determine if something is a gene or not. I'm not going to go over them one by one here. I'd like you to read over them before you start. So you can pause this video, read over these guiding principles. They might not make a whole lot of sense to you, that's fine. After we annotate our first gene, we will come back to the guiding principles and see, does this annotation go along with these guiding principles? So if we go past that, this takes us to chapter 8. Chapter 8 here is where we start actually annotating our genome. So this is our actual annotation here. And it gives you an overview of what this means. It gives you the items that you should probably have on hand. The first is your annotation file open in DNA Master, which we do. So that's our file that we downloaded from eClass and just opened. A printout of the guiding principles of bacteriophage annotation. That's pages 64 and 65 in your annotation guide. Famirator, we will open this as we need it. GeneMark, we will open this as we need it. And we generally don't need number 5 or 6 here. So we're going to go ahead and we're going to start annotating a gene. Now, this section here, 8.3, this is something that you might want to keep on hand. This is a decision tree for your annotation process, which means you may want to use this each time you look at a gene when you're trying to decide what the next step should be. So in this, blue boxes are decision points. So you're going to decide what you should do there, and it'll tell you where in the annotation guide you can find information about making that decision. Most of it is in section 8. The purple boxes are action points where you implement something. So when you get to things like deleting a gene, changing a start, adding a gene, these are in section 9. We will kind of deal with those as we get to there. So the vast majority of the time you're going to start right here with protein coding genes. Almost all of the annotation begins with protein coding genes. So the first thing that we want to ask is this gene call supported by strong evidence? So we are going to start by annotating gene number 3. So if we go back to DNA Master, if we go to Features, I'm going to widen the feature list by right-clicking on that box. We're going to start annotating this gene here, gene number 3. And I'm going to click on it just so we have all the information available. I'd also recommend go to DNA, click on Frames, and click on ORFs, because this is going to give us a visual representation of all of our genes, including gene 3. And I'm going to zoom in once, and then I'm going to click on this left arrow focus a little more on gene 3 here, and then I'm going to zoom in one more time and hit the left arrow. So this is gene 3. You know where it ends because this is the stop codon. So this is the end of gene 3, and right next to that is the beginning of gene 4. This is the start codon that the audit annotation chose. So if we go back to our decision tree here, the question is this gene call supported by strong evidence? And we're going to look at the different evidence that we use in section 8.41 to determine is it an actual gene call. Now, one other thing you might want to do is you may want to open your lab notebook as well as the Google Sheet for Octavius Rex, because we're going to put some of this information in as we go along. So this is my viral discovery wiki. This is the Google Sheet for Octavius Rex that is tethered to the wiki notebook. So we've got DNA master here. So let's go down to section 8.41. We're looking at protein coding calls. Non-protein coding calls, we will deal with those as we get to them, but the vast majority of features that you're going to investigate are these protein coding genes. So this is what we're going to practice with a number of genes. The first question is the designation of this open reading frame. So remember, an open reading frame is from a potential start codon to a stop codon. So this is the open reading frame for number 3 here, gene 3, just like that. Is it well supported? So we go to the Features tab, we pick, they're looking at gene 5, we picked gene 3. The first thing we look at is this box here, right down here. It's going to tell us what the original call was. So here it says original glimmer call at base pair 1888. So the notes in here are going to tell us whether the 2 prediction programs predicted a gene. Now, I'm actually going to open a file here so I can type this out because I want to make sure that you see this. If it says glimmer predicted a gene at whatever number, what that means is that both glimmer, and glimmer is one of the gene prediction programs. There are 2 gene prediction programs that we run in DNA Master. Both glimmer and GeneMark chose the same start site. So if these are 2 gene prediction programs. If they both chose the same start site, it will say glimmer predicted or original glimmer call at, so it says original glimmer call at whatever number. That means both glimmer and GeneMark called that. If it says original glimmer call at a number, and then it says original GeneMark call at a number, what that means is that glimmer, and again these are just 2 gene prediction programs, and GeneMark chose different start sites, and what we'll have to do is determine, are one of them right? If it says only original GeneMark call at, and it doesn't say anything about glimmer, it means that GeneMark called this a gene and used this start site. Glimmer did not. Those are the ones that you're generally going to see. So most of them will likely be this top one here. It'll say original Glimmer call at. What that means is both Glimmer and GeneMark chose that start site. You will sometimes see this one as well, where two different start sites are chosen. Usually if one calls it a gene, the other will call it a gene. If we come across examples other than these, we will deal with them as we come along. So here we see original Glimmer call at base pair 1688. So what this means is, and I'm gonna put this in my lab notebook because it's a good place for me to keep track of it. So I'm gonna go to edit, and I'm gonna go in here, and this might be the beginning of my annotation. And I'm going to make this a little bigger. And we're going to start with gene 2, or sorry, gene 3. And Glimmer and GeneMark both called at 1888. This is the location in the genome that it predicted a start site. So right away, yeah, it's good evidence that there's a gene if both of these predict it. So, and what I'll do is I'm going to put for this gene what this data is supporting. So here we're asking, does Glimmer and or GeneMark call this a gene? So the next question is there what's called coding potential here? So coding potential is when these prediction programs look at a sequence and determine, does this have the ability to make a protein? To get to this, what we use for this, you're going to need to go back to eClass, and you're going to have to go to the Octavius Rex phages DB site right here. And we're going to click on that. And that's going to take us to the page for Octavius Rex on phages DB. And there are 3 different GeneMark programs that you can see here. This one, and this one. We're only going to use one. We're going to use this one called Run GeneMark SMEG. And what this program does is it takes the sequence from our virus and it compares it to how Mycobacteria smegmatis, which is the host, typically uses sequences for genes. And it says, based upon how smegmatis determines if something is a gene, would this be a gene from our viral genome? So we're going to click on this here. And I'm actually going to go back and I'm just going to open this in a new tab. So I'm going to right click, open it, new tab. And it's going to give us something that looks like this. This is what's called the GeneMark output. So if we want to determine if there is coding potential, we're going to scroll down, and for each of these pages there are 6 reading frames. 1, 2, 3, 4, 5, and 6. And below each of these will be the nucleotide position. You can see that this page goes from 0, each of these hash marks is 100, from 0 to about a little after 2100. And then it picks up on the next page, 2200, 2300, 2400. So in order to find this gene, we always go to the stop codon and look for where that gene is. We know that this is on the forward strand because it's in +1, +2, or +3. Now, these numbers here, +1, +2, +3, don't necessarily correlate with +1, +2, +3 here. But if it is in a plus up here, so if it is +3 or +2 or +1, it will be in one of the top 3 of these here, because it's saying it's on the same strand. So we're going to look for somewhere around 1888. We're going to look for a stop codon. So 1888 is going to, so this is 1900, so not there. Aha, there's our stop codon. So the question is there coding potential in this region? And anytime you see these peaks, the area under these peaks is coding potential. So because there's a lot of area under these peaks, in fact, there is coding potential here. So as long as you have peaks here, we would say, yes, there's coding potential. So we're going to go to our notebook and say, yes, gene mark shows coding potential. And that's what it's looking at on page 69 here. So another question, and we're not looking at the start site yet. We're still just determining, is this a good gene? We will come back in a minute to look at, is this a good call for a start site? So the next thing we want to look at is BLAST. So how many other microbacteriophages have this gene? And again, we'll come back to the start site. We just want to know, are there other phages that have this gene? So we're going to go back to this window here. And over here, we're going to click on this tab that says BLAST. Like this. This list here are different matches. So you can scroll down here and it give you the score and the match. So in the Mycobacterium phage whose name is Leo, there is a match. And if you want to look at the match, you can click down here on this tab that says alignment, and it'll actually show you how those two amino acid sequences align. So this is the protein that is predicted in our virus, the top row. And sorry, I'm trying to get back to number 1 here. There we go. And the bottom row is the amino acid sequence of the match, Leo. And if you go to HSP data, it's going to give you the E value, which is 0, which is really good. It's a really good match. It's 100% identical. You can see there are other ones from other phages as well. Angel, Halo, Leafy, Tahira. These all match up really quite well. So the question is, are there other genes that match up in other phages? And the answer to that is yes. So is there a match with other phages for this gene? And the answer is yes. Many phages show a BLAST match. So this is what they're showing on page 70. The next thing we ask is, what about the length of the gene? Is it long enough for us to consider it likely to be a gene? So if you read through the annotation, the guiding principles, most genes are going to be about 150 base pairs or longer. There are some that will be shorter, but if they're longer than 150 base pairs, that's pretty normal. Here, 1,515 base pairs. Pretty long. So is the gene sufficiently long? Yes. It's 1,515 base pairs. And then the last thing that we want to ask, and this is where you can see the length as well. If you go back to description, 1,515 base pairs. And again, as I'm going through this, I highly recommend reading what they talk about in the annotation guide. I'm not going to read these word for word, but it's good for you to take a look at that and read that. The last thing is there only one gene called in this region of DNA? We haven't generated a DNA master map, but we will, and we will bring this to class. If we look at the frames here, you can also see, looking at, and I'm going to zoom out by clicking this minus magnifying glass, you can see in this region there's only one gene that has been called. And this, in viral genomes, it's very rare to see genes that overlap significantly. They will oftentimes overlap a little bit, but it's rare to see them overlap significantly. And if you look at the guiding principles, it will tell you what that number is. So, and like I said, you don't have to put these questions in here. This is just for a guide for this walkthrough right here. So, is there only one gene called in this region? Yes. So, based upon this, based upon all this data, we can say this highly supports. Now, because we don't like to use the word proves, it doesn't prove anything. This data highly supports this being a gene. We just came to the conclusion that we're pretty confident that this is that the data highly supports this being an actual gene. So if we go to our decision tree, which is back up here, is this gene call supported by strong evidence? Yes. So the next question is the called start site the best possible choice? So we need to go to section 8.42 to determine that. So is the designation of this gene, this is kind of the decision we just made. Most gene calls will be yes, ours was yes, so we're going to say, let's continue to this section here, 8.4.2. Is the called start site for this gene the best possible choice? So there are a few different lines of evidence we want to look at. The first one is, does the predicted start site include all of the coding potential? So again, the coding potential by this program GeneMark tells us which part of the genome it thinks is likely to be a gene. Now, the picture showing here shows a red and black lines. Ours only has the black line. But if you imagine this, an example, this is a gene, a potential gene here. One potential start site was here, one potential start site was here. So we'll call this here A and B. So A here, since this is the 5' or the left side of the gene, everything here is, all of the coding potential is to the right of this, which means that everything downstream of our start site is where the coding potential is. So this would be a good choice for this. This here does not include all of the coding potential, because if this was our start site right here, so I'm just going to try to draw a line up here like this. So, sorry. If this is our start site right here, then there is some coding potential to the left of that, which means that there is a good chance that that is part of a gene. So let's go back and look at our gene here. So remember, this was the stop codon, or stop site 18888, and we want to find the start site that is called. So if we go back to our gene here, gene 3, or I'm sorry, the stop site was 3402 here. So previously, I apologize, previously I was looking at the wrong gene. 3402 is right here. So this here is the coding potential region for this gene. So you can see this gene has a lot of coding potential. So again, previously when we were determining if it was a gene, I was looking at the wrong part. This is gene 3. This is the end of gene 3 right here, 3402. There's a lot of coding potential. The question is the start site that's chosen, does that include all of the coding potential? So you can see this is in frame 1, so we have to go back up to the previous page. And this is the beginning of that gene. The start site that's called for that is 1888. So if we go to 1888, so this is 2000, 1900, 18888 is probably around here. So since all of the coding potential is to the right side of this, so if we kind of draw a line here, we were to draw a line right here, all the coding potential is to the right. So all of the coding potential is included in the part of the gene that was predicted. So here we would say so for the question, does the currently predicted start site include all of the coding potential in the gene mark output? And here I would say, yes, it does. And I know these fonts are different, but I'm just copying and pasting some things. So the start site looks good because it takes into account all the coding potential. Did both Glimmer and GeneMark call the same start site for this gene? So remember, we were talking about this. If it says just original Glimmer call at, it means that they called the same start site. Here it says original Glimmer call at base pair 18888. So what that means is, yes, both of these prediction programs chose the same start site. So did Glimmer and GeneMark agree on the start site for this gene? Yes, they did. Number 3, is the predicted start codon the longest possible open reading frame without causing excessive overlap? So remember, if you go back to pages 64 and 65, they talk about the guiding principles of annotation. And they say genes do not overlap by more than a few base pairs, although up to 30 base pairs is legitimate. So the question here is the predicted start codon the longest possible ORF? So is it make the longest open reading frame? Because in general, genes are usually the longest possible open reading frame without excessive overlap. So to answer that, we have to go back to DNA Master, and we have to go to our frames. And if you closed out your frames, if you want to open it again, you go to DNA, Frames, and then click ORFs. And we're just going to zoom in around the beginning of gene 3 here. So I'm going to press zoom, and then the left arrow, and then zoom again, and then the left arrow. So is this the longest possible open reading frame without significant overlap? I'm going to zoom one more time. So you can see there might be a little bit of overlap here. The next possible start site is way up here with a big overlap. Here. So if we actually look at, if we go back to this window here, how long is this overlap? So the previous gene ends at 1891. This gene begins at 18888. That's an overlap of 4 base pairs. It turns out that an overlap of 4 base pairs is ideal. So I'm going to write this down so we remember it. A 4 base pair overlap between 2 genes is very good. And we'll talk about why that is in a little bit. So this is usually indicating that this is a very good start site call. And again, we know that because the previous gene ends at 18891. This one starts at 18888. So nucleotide 888, 889, 890, and 891 are used by both genes. So it's a 4 base pair overlap. So is this the longest? Is the predicted start codon the longest possible ORF without causing excessive overlap? Yes, it is. So the next question. So now we're on to page 73. Does the start site match other starts for similar genes in GenBank and the microbacteriophage database? So this is where we have to use BLAST. And we typically use 2 different BLAST programs for this. And it's good to look at both. So we can use the NCBI BLAST and we can use the Phages DB BLAST. Now, what this is looking at is for other genes, other genomes that have been annotated, do the start sites match up? Remember, these other genes that have been annotated, they could be incorrect, so don't be overly enthusiastic. But if there are a lot of ones that use the same start site, then it's likely that they're going to be correct. So first we can look at the NCBI BLAST. This is the one that was done in DNA Master. So we go to DNA Master here, where our main window, and over here we're going to click BLAST. And remember, these are our hits, so we're going to start at the top one. The top hit uses, so whenever you see 1 and 1, it means they're using the same start site. So the top hit here uses the same start site for this gene. This hit is very close. It's off by 1 amino acid. The third one, same start site. The fourth one, now these are starting to differ a little bit, but you can still see a lot of them. I'm just scrolling down. A lot of them are using the same start site. We can also look at the Phages DB BLAST. So if we go here to our browser and type in Phages DB or I'm just going to do Phages DB BLAST, it'll be our top hit. We're going to use the program, and we want to go to Protein BLAST here. So make sure that you're doing a local Protein BLAST. We're going to put in the sequence of this predicted gene. So to do that, we're going to go back to DNA Master. So we're on the BLAST page here. If we click Product, it's going to give us our amino acid sequence. So remember, we've been in description, we've been in sequence. Product is going to give us our amino acid sequence. We're going to copy that. So I'm just going to highlight it and then edit, copy. And then I'm going to go back to here, to my browser, and I'm going to paste it and click BLAST. And this is similar to the blast results you saw before. However, the difference is you're just blasting one gene here instead of the entire genome. And it's going to tell you which phages have genes that match up very closely to it. You can see they're listed right here. We're going to scroll down until we see the alignments, and then we can look at the alignments. And it's going to show us how closely they align. So this one, it starts there. The start site is very similar, 1 versus 2. Same thing here, 1 versus 2, 1 versus 2. So here, 1 versus 1, which means exactly the same. 1 versus 1, 1 versus 1. So it looks like these are all using either the same or a very similar start site where it might be off by amino acid of one amino acid. So this can give us pretty good confidence that this is in fact our start site. So the question here is, does the start site match other start sites for similar genes? And here, or in our notebook, we're going to say yes. And we may want to show an example of ones that are, this can be from NCBI or from the other the Phages DB database, but it's good to give an example of which those alignments are. So this one here, it's hard to see the name of this particular one. This is, so for example, see if we can't make this a little bigger here. So GUO1, so GUO1 is the same, and Tahira is the same. So if we just give 2 or 3 that use the same site, it backs up our prediction. So we've looked at, does the start site match other starts? And again, I would read through these parts of the annotation guide as you're going through it. I'm not going to read them directly, but you should take a look at these. The next one, using Starterator to evaluate gene starts. So we're actually going to, before we do that, we're going to use what's called Famerator, because we have to use Famerator before we can use Starterator. So the details for how to use Famerator are on page 49 of your annotation guide. There is an explanation of what Famerator is. We will go over Famerator more in class. It's about 10 pages of information about this. I'm just going to show you how to pull up the genomes in Famerator. Now, how do we get to Famerator? Famerator is on, you're going to get to through the browser. So you're going to go to this website here, Famerator, PHAMERATOR.org. And you're going to have to register, and it only takes a couple minutes. Register, make an account, it's free, and it's going to bring you to a page that looks like this. This is what we use to compare different genomes. So we're going to, once you register, go through the process, get here, we're going to click on Genome Maps. And every single sequenced phage from this project is in Famerator here. So now we have to tell it which phages we want to compare. And generally, we want to compare to phages that are very similar to ours, because there's more likely that those genes are going to be conserved. So which genes do we compare it to? Well, this is where we're going to go back to our notebook and the results of our original BLAST search. So if I save these in notes here, and these were all the genomes that were very similar. So we would love to compare it to some of these genomes. And if you remember, these were all cluster G genomes. So we're going to look at the genomes, we're going to go to FAMReader and we're going to look for the cluster G genomes. So I'm going to go back to FAMReader. I'm going to click on these three lines and it gives me a plus or an X. I'm going to click on the plus and that's going to expand each of these boxes so I can see the names. These are all different sequenced phages. And I'm going to scroll down to G. There we go, here we go, here we go. And I'm just going to unclick everything here. Yours won't be clicked. And you can see our phage is G1 cluster. And you can see Octavius rex, it says draft because it hasn't been annotated yet. So the draft ones are ones that have been sequenced but not annotated. We want to compare ours to sequences of genomes that have been annotated. So what we'd like to do is look at this list of phages and find ones that are similar, probably in this group here. Find ones that are similar that don't say draft next to it. So you might want to take a second, see what you find. So you can go back and forth between this and here and find some that are who have genomes that are very similar to our phage's genome, but they've been annotated. You can pause this video while you do it. Now, when I did this, I chose Ghomashi and Frosty24 because they're both very similar. So Frosty's right here. Sorry, Frosty, Ghomashi. So we usually, comparing three of them is fine. And then we also have to click Octavius rex draft. So we've selected three genomes. And the instructions that you have in your annotation guide are somewhat right, but also somewhat not, because this year is the first time we have an online version of this program. So like I said, we will work with you to figure out all the different functions of this, but for now just follow along with what I'm doing. So we've picked our three genomes here: Ghomashi, Octavius rex, Frosty. I'm going to scroll back up to the top. And instead of select phages, I'm going to click on view map. And you're going to get something that looks like this. This is a visual representation of the Frosty genome. This is a visual representation of the Ghomashi genome. This is a visual representation of our genome. Now you can see, depending on the settings, some of the Ghomashi and the Frosty24, their genes have names over it. And that's because those genes have been annotated and these functions have been assigned to them. Ours does not. Since ours is unknown, it actually works best to put ours in the middle of the other two genes. We can do this by putting our cursor over Octavius rex draft when you see the plus there, and just dragging it up above Ghomashi, and it's going to put that one in the middle. So what this is showing is it is comparing the sequence between these different genomes. Purple is the comparison of the nucleotide sequences. So anytime you see purple, it means they're very similar, and it will give you an e-value for the difference between the genomes. So you can see an e-value of 0, e-value of 0, how similar they are, just about 100%. These are very, very similar genomes. The boxes are the individual genes, and the colors represent these are different proteins. So we're looking at protein 3, or gene 3. So this is gene 3. In Frosty, gene 3 is a portal protein. In Gamashi, it's just portal. This all depends on how people annotate it. And here we haven't figured out what it is yet. You can notice that these all have the same number above them. This is the FAM number, PHAM. So these different genes get lumped into what we call families, PHAMILY. If they're very similar to each other, we can say they're in the same family. And here, these three genes are very similar. You can see they're the same size. They all start at pretty much the same location. So we can use Famirator to compare our different genes. What we need is to write down this number, 7340. This is the number of our gene FAM. This is how we're going to identify our gene, and I'm going back to the annotation guide here, in this program called Starterator. So we're going to use Starterator to find this gene numbered, or this FAM numbered 7340. Now, Starterator is another program that we can run in another operating system, but instead of using another operating system, we can just download a PDF of what we would have used. So for this, we need to go back to our, and we'll talk more about Famirator as we go along, but the main reason we used Famirator right here was to compare the same gene between our the other ours and the other two genomes, and to figure out what FAM number this is, 7340. So next we need to go to the web page that has all the Starterator information. You need to go to this website up here, phages.wustl.edu/starterator, and it'll bring you it'll bring you to this directory here. This is all of the different PDFs for all of the different FAMs. I'm just going to use my find function, Command F, and I'm going to type in, because you can see that here they're in numerical order. So instead of scrolling down to 7,000, I'm going to type PHAM 7340. And it's going to give me one hit, and here it is. This is the FAM report for this particular gene that we're annotating. And I'm going to click on that, and it's going to download it. And you're going to see something that looks like this. Now, right away, this is going to look scary. Now, in your annotation guide, like I said, I would read through pages 48 to 58 just to give you some background on Famirator. We will go into that more. Starterator is on page 59. This is the result of a Starterator analysis. So this program can be a little confusing. I'm going to walk you through it the best I can.